purpose of our study was to really look for the first time in high resolution at the epigenomes of both embryonic stem cells and induced pluripotent stem cells. What we learned from this study was that embryonic stem cells and induced pluripotent cells overall look fairly similar. But what we found is there are differences, and these differences can be identified in different types of induced pluripotent stem cells. And it differentiates their epigenome from embryonic stem cells. So this allows us to identify a signature of the reprogramming that hasn't been fully completed in induced pluripotent stem cells. This IPS signature will allow us to try to understand the mechanisms now that differentiate ES and IPS cells. Now that we have identified those locations, we can begin to carry out experiments to ask what's missing in the reprogramming events. Why aren't these IPS regions reprogrammed properly? The epigenetic signature is there in many different types of IPS cells, so we know that it's a parameter that happens every time you carry out the assay. And so it provides a way of distinguishing ES and IPS that we didn't have before.